Good morning. My name is Deja Crossmith. Today we'll be diving into the fascinating world of basic cellular genetics. We'll explore how genetic information is not only stored and replicated, but also how it's interpreted to create the proteins that define who we are. On a cellular and organism level, our goal is to move beyond memorization and focus on connections between molecules, mechanisms, and meaning. By the end of this session, you should be able to describe the structure and replication process of DNA, explain gene expression through transcription and translation, and analyze how the genetic code contributes to organism uniqueness. These are foundational principles, but we'll approach them through a critical lens, thinking about how and why these processes are evolutionarily conserved and biologically efficient. Understanding genetics isn't just for future biologists. It underpins nearly every biological discipline, from diagnosing inherited disorders to designing CRISPR therapies to understanding antibiotic resistance. Genetics touches forensics, agriculture, even the ethics of cloning and privacy. But at its core, it's about identity. Why do two siblings look different? Why does a single mutation changes a protein so drastically? These aren't just textbook questions, they're questions of life. Start at the molecular level. DNA, which is deoxyribonucleic acid, is a double helix composed of two antiparallel strands. Each strand is a polymer of nucleotides, each containing a phosphate group, a deoxyribose sugar, and a nitrous base. You know the pairing, A with T and C with G. One of the ways that I always remember this is that there are apples in a tree and cars in a garage. Hopefully that helps you. But why do we need to know this information? Hydrogen bonding provides specificity, but base stacking, interactions between adjacent bases, gives the helix its stability. The 5 to 3 directionality is critical, especially for replication and transcription. The sequence itself holds the code, but the structure, its ability to coil and loop and compact, is just as essential. DNA replication is semi-conservative. Each new double helix contains one original strand and one newly synthesized one. Helicase unwinds the helix. DNA polymerase adds nucleotides in a 3 to 5 direction, and the ligase seals the gaps between the okeanasi fragments on the lagging strand. But it's more than just mechanics. Replication is timed to the cell cycle, proofread for fidelity, and remarkably fast. Because you think about 50 nucleotides per second. Think of the implications of this. A single uncorrected error can lead to a mutation which may be silenced or may spark disease. Gene expression is the process by which the instructions in DNA are converted into a functional product, typically a protein. The central dogma, DNA to RNA to protein, is conceptually simple but mechanically complex. Transcription is highly regulated and involves promoter recognition, initiation, elongation, and termination. Translation requires precision decoding of codons by tRNA anticodons. And here the key, gene expression is responsive. Environmental cues Developmental stages, even circadian rhythms affect which genes are on and off. This isn't a static system, it's dynamic and contextual. In eukaryotes, transcription occurs in the nucleus. RNA polymerase II binds to the promoter region, aided by transcription factors. It synthesis a complementary strand of mRNA, substituting uracil for thymine. Pre-mRNA is spliced to remove introns, and a 5' and poly A tail are added for protection and export. One common misconception is that genes are contiguous code units. They are not. Alternative splicing can yield multiple proteins from the same gene, increasing proteomic complexity without expanding genome size. This genetic efficiency this is genetic efficiency at its finest. Action happens in the cytoplasm at the ribosome. mRNA mRNA codons are read in triplets, each specifying an amino acid. tRNA molecules bring amino acids to the ribosome, matching codons via their anticodon loops. The ribosome links the amino acids with peptide bonds, forming a growing polypeptide chain. This process continues until a stop codon signal releases. But think deeper. How do we ensure accuracy? The redundancy of the genetic code, proofreading, amino acid tRNA syntheses, and ribosomal fidelity all minimize error. A single nucleotide mistake might lead to a misfolded protein, or worse, a non-functional one. The genetic code is universal. Nearly all organisms use the same codon to amino acid assignments, which supports the theory of common ancestry. The code is also redundant. Multiple codons encode the same amino acid. This provides a buffer against mutation. Silent mutations don't always alter protein products. However, mutations in critical regions like start codons or splice sites can be catastrophic. Missense mutations may substitute one amino acid for another. Nonsense mutations create premature stop codons. The code is robust yet delicate. How does this molecular machinery make you? You. Variability arises from SNPSs, which are single nucleotide polymorphisms, and larger structural variations. During meiosis, crossover and independent assortment shuffles alleles, generating gametes with unique combinations. Epigenetics modifications like DNA methylation further regulate gene expressions without changing the DNA sequence. These combinations of genotypes and epigenetic influences give rise to phenotypes. Even identical twins differ in gene expressions as they age. In genetics, individuals is not just encoded, it's orchestrated. Teach this content to AP, undergraduate, or dual enrollment students. It must go beyond definitions. Such tools like models of 3D DNA or interactive codon games enhances comprehension. Case studies such as the CFTR gene mutation in cystic fibrosis illustrate molecular principles in clinical contexts. Students can extract mRNA sequences and use codon tables to predict protein structures. This encourages them to think outside of the box, but also Certain online tools like the NCBI BLAST or UCSC Genome Browser can help with active learning, which pushes them to question, analyze, and apply essential skills to become the next generation of researchers. To dig deeper, reading the ENCODE project, which mapped functional elements of genomes, explore how CRISPR-Cas9 revolutionized gene editing by targeting specific sequences, or examining how RNA sequencing reveals which genes are active under different conditions. For emerging scientists, these tools and databases are gold mines. Access to primary literature such as Nature Genetics or Cell can also train them how to critically evaluate genetic research. To recap, DNA structure and replication ensure fidelity across generations. Gene expression translate code into function, dynamically regulating by context. The genetic code, though universal, is the blueprint for biological diversity. Together, these systems explain not only how cells operate, but why organisms are both similar and unique. That's the power of molecular genetics. It explains the unity and diversity of life.